As far as known as proteorhodopsin functions like bacteriorhodopsin, except that several different spectral forms exist, each tuned to the absorption of different wavelengths of light. Proteorhodopsin, as a mechanism for energy conservation in marine prokaryotes, makes good ecological sense, since concentrations of dissolved organic matter in the oceans are typically very low, and thus strict chemoorganotrophically would be a different lifestyle. Methane-producing archaea methanogens. Methanobacteria, methanocaldococcus, and methanosarcina. A large number of eukaryotic produce methane, CH4, as an integral part of their energy metabolism. Such as organisms are called methanogens, and the process of methane formation and methanogenesis. Methane was first discovered as a type of combustible air by the Italian physicist Alessandro Volta, who collected gas from marsh sediments and showed that it was flammable. The Volta experiment can be easily reproduced if methane trapped in freshwater sediments is collected and carefully ignited. In late chapters, we will study the biochemical, unique, and amazing complex process of methane formation, and then learn how methanogenesis is the terminal step in the biodegradation of organic matter in many anoxic habitats in nature, such as the swamp shown. The major sources of biogenetic methane are listed in this table. Diversity and physiology of methanogens. A wide variety of morphological types of methanogens have been described. Their taxonomy is based on a phenotypic as well as phylogenetic comparative 16-sulfur rRNA sequencing analysis, with several orders being recognized in taxonomy. An order contains a group of related families, each of which contain one or more genera. Structurally, methanogens are prokaryotic cells that show a diversity of cell wall chemistries. The latter include the pseudo-peptidoglycan walls of Methanobacterium, species of relatives. The methanochondroidium, so named because of its structural resemblance to chondroitin, the connective tissue polymer of vertebrate animals. Walls, methanosarcina, and relatives. The protein of glycoprotein walls of methanocaldococcus and the methanoplanus species respectively, and S-layer walls of methanospirillum. Physiologically, methanogens are oblique anaerobes, and strict anoxic techniques are necessary to culture them. Depending on the species, cultures of methanogens can be established in a mineral salt medium under atmosphere H2 plus CO2 in the ratio of 4:1. See the reaction shown later. Or in complex media. Most known methanogens are mesophilic, although extremophilic species growing optimally at very high or very low temperature or at a very high salt concentration have also been described. Substrates for methanogenesis. At least 11 substrates have been shown to be converted to methane by pure cultures of methanogens. Interestingly, these substrates do not include such common compounds as glucose and organic or fatty acids, other than acetate or pyruvate. This is not to say that a compound like glucose can never be converted to methane. We will discuss in Chapter 19 how in cooperative reactions involving methanogens and other anaerobic bacteria, virtually any organic compound can be converted to methane, CO2, and CO2. 3 classes of compounds make up a list of 10 methanogenic substrates shown in Table 13.6. These include CO2, type substrates, methyl substrates, and acetotrophic substrates. The first class includes the important substrate CO2, substrate which are reduced to methane using H2 as electron donor. Other substrates here include formate, which simply CO2 plus H2 is combined form, and CO carbon monoxide. The second class of methanogenic substrates are methyl group substances, using CH3OH as a model methyl substrate here. The formation CH4 can occur in either of two ways. First, CH3OH can be reduced using an extremely electron donor such as H2. CH3OH plus CH2, CH4 plus H2O, delta G to the power 0 equals negative 113 kilojoules. Alternatively, in the absence of H2, some CH3OH can be oxidized to CO2 in order to generate the electron needed to reduce other molecules of CH3OH to CH4. 4 CH3OH, 3 CH4 plus CO2 plus 2 H2O, delta G to the power of 0 equals negative 319 kilojoules. The final methanogenic process is the cleavage of acetate to CO2 plus CH4, called the acetotrophic reaction. CH3COO negative, power negative, plus H2O C2CH4 plus HCO3 negative, delta G to the power of 0 equals negative 31 kilojoules. Only a very few methanogens are acetotrophic, yet experimental measurements of methane formation in methanogenic habitats such as sewage sludge have shown that about 2/3 of the methane formed originates from acetate and 1/3 H2 plus CO2. Thus, although lacking in terms of known diversity, acetotrophic methanogens are very ecologically significant in nature. As can be seen by inspection of each of the above reactions, they are all exergonic and can thus be used to synthesize ATP. We reserve until Chapter 17 the biochemical details of methanogenesis and only note here that the process is coupled to protein motor force, formation, ATP synthesis, normal chemoosmotic mechanism, substrate level of phosphorylation typical of fermentative bacteria apparently does not occur in methanogens. Methanocaldicoccus janusci as a model methanogen archaea. As will be discussed in section 15.3, the complete genome sequence of the hyperthermophilic methanogen Methanocaldicoccus janusci has been determined. Megabase circular genome of M. janusci contains about 1,700 genes, and from sequence analysts, gene encoding enzymes of the methanogens and several other key cell functions have been identified. Interestingly, the majority of M. janusci genes encoding functions like central metabolic pathways and cell division are similar to those in bacteria, while most of the genes encoding core molecular processes like transcription and translation more closely resemble those of eukaryotes. These findings support for evolutionary tree of life that shows the domain archaea positioned between the domain bacteria and eukarya. However, sequence analysts also showed that nearly 50% of janusci genes have no counterparts and known genes from bacteria eukaryotes, suggesting that there may be many new cellular functions encoded in archaea DNA that have yet been discovered. Thermoplasmatales, thermoplasm, ferroplasm, and pyrophyllus. Thermoplasm and pyrophyllus. A phylogenetic distinct line of archaea contains 3 thermophilic and extremely acidophilic prokaryotes. These organisms are among the most acidophilic of all known prokaryotes, and in the case of pyrophyllus, even capable of growth below pH 0. They also form their own order of prokaryotes within the eukaryota. Euryarchaeota. The thermoplasmatales. We begin with the description of the mycoplasm-like organism thermoplasma. Thermoplasma and ferroplasma, cell wall-less archaeans. Thermoplasm and the chemoorganotroph and grows optimally at 55 degrees Celsius, and pH 2 in complex media 2 species have been described. Acidophyllum and T. volcanium species of thermoplasm are facultative aerobes growing either anaerobically or aerobically by sulfur respiration. From a morphological point of view, thermoplasma resembles the microplasms, in that it lacks a cell wall. Most strains of thermoplasma have been obtained from cell heating. Coal refuse piles. Coal refuse contains coal fragments, pyrite, and other organic materials extracted from coal, and when dumped into piles and coal mining operations tend to self-heat by spontaneous combustion. This sets the stage for growth of thermoplasm, which apparently metabolizes organic compounds leached from the hot coal refuse. The second species of thermoplasm, the T. volcanium, has been isolated from the salpeter fields throughout the world and are highly motile by multiple flagella. To survive the osmotic stress of life without a cell wall and to withstand the dual environment extremes of low pH and high temperature, thermoplasm has evolved a cell membrane of chemically unique structure. The membrane contains a lipopolysaccharide-like material, referred to as lipoglycan, and mycoplasms, consisting of a tetraether lipid with mannose and glucose units. The molecular constituents and major fraction of the total lipid composition of thermoplasm. The methane also contains glycoproteins but not sterols. Together these and other molecules render the thermoplasm membrane stable to hot acid conditions. The genome of thermoplasm is of interest. Like other mycoplasms, thermoplasm contains a small genome. In addition, the DNA is complex with highly basic DNA-binding protein that organizes the DNA into globular particles resembling the nucleosomes of eukaryotic cells. Discuss the arrangement of DNA in eukaryotes. This protein